to more microenvironments and studies those. So thank you so much for being here and for showing your work.
Thank you very much for the opportunity. I too have had a great time listening to everyone else's work, and look forward to telling you a little bit about what we're doing. Some of the same themes that we've heard about for the past day or so will come up again in this presentation. So as Stefan mentioned, what I'd like to do is give you a broad overview of some of the work that is going on in our Link Center out at OHSU. The general goal of our center is shown schematically here in this cartoon. The idea behind our center is to try to understand how intrinsic and extrinsic factors coordinate to modulate cellular phenotypes. So shown schematically at the bottom are some of the sorts of external signals that we think are particularly important to study, both in the context of cancer as well as in normal cells. We know in this particular case, actually throughout, I will use a little bit of jargon. ME in this particular case is microenvironment. MEP is microenvironment perturbation. In the parlance of links, we think a lot about perturbing cellular systems. And in particular, what we're interested in studying the extracellular signals come from, for instance, neighboring cells such as fibroblasts, immune cells, as well as non-cellular components. We know that within the human body, there's no such thing as a cell sitting in isolation. And so what we really want to do is think about how cells respond to the many factors they're exposed to continuously. Additionally, we know that internal to cells, in particular cancer cells, there are many intrinsic changes that happen. We just heard in the preceding talk about changes that may happen over the course of one's life. And these two sets of factors, the extrinsic as well as the intrinsic, integrate to modulate cellular phenotype. And we're particularly interested in the phenotype shown here on the right-hand side: proliferation, apoptosis, differentiation state, therapeutic response, and we've become increasingly interested in the phenotype of migration. And I have to do these to show you later in my presentation. So as I mentioned, microenvironmental signals originate from many sources, and we know they also vary tremendously across different tissues in the body. Shown schematically on the left-hand side is a little example. If we imagine a ball of cells in the middle, that red ball of cells up there, this, in the context of cancer, these cells, or even in normal tissues, may be exposed to many different signals that arise from different cells within their local neighborhood. So those are shown schematically on the bottom here. So there are stromal cells, lymphatic cells, local ECM, and so on. And what we can do is actually identify different proteins of each of these classes and begin to try to think about how they may modulate cellular phenotypes. Additionally, if we look at large-scale publicly available data sets, such as GTEx type data sets, we can clearly demonstrate that these types of signals vary tremendously from tissue to tissue across the body. So what I'm showing you in the heat map are the expression of these different proteins in brain, bone, liver, and so on. I've chosen these particular tissues to show you because we know in the context of cancer, these are actually incredibly relevant sites to think about. One imagines a primary disease that may originate, for instance, in a patient's breast, ultimately leads to metastases in different tissues of the body, such as those I've listed here. And those local neighborhoods then are tremendously different. And so if we really want to think about how to accurately treat patients with metastatic disease, we need to understand how cells respond to these different environmental components. So in order to study these sorts of proteins, what we've done is to develop what we call a microenvironment microarray, or a MEMA. And this is a high throughput platform to study combinatorial microenvironmental signals. And so what we do is actually fabricate these in our lab using a robotic printer shown here on the left-hand side. We print down into 8-well plates. The format is actually fairly flexible. In this case, I'm showing you one array that we print down, about 700 spots. In high resolution in the middle there, you can see there's spots about 400 microns. These form little growth pads upon which cells can grow. They grow only on those spots and effectively are treated by the extracellular matrix proteins that we print down. After several days, we can fix and stain these cells. The assay itself is shown here on the far right-hand side, where we're printing down ECMs. We can then flood on different ligands, and so this leads to this combination that I mentioned. What we have shown over the course of several years is that this platform has broad utility and can be used to study many different cell types, which I'm showing here. In each of these cases, the cells grow in this little circular formation because they're growing only upon these growth pads. And this sort of high content image data is the type of data that Anne very beautifully described yesterday morning during our presentation, her presentation rather. We spent a lot of time working on generating workflows to both generate these data as well as manage and analyze the high content image data. So shown schematically here is the workflows for MEMA as well as the image management infrastructure we've stood up. And we put a lot of effort into the analysis and QC of these sorts of data, and also are working on sharing it through various websites. Our website on Synapse, as well as working with the DCIC to send these data out to the Links project site. In terms of the image management, that's actually been one of the biggest challenges. This truly is a big data problem. We think about big data oftentimes as sequence data, but in fact, we are generating reams and reams of image data that we've had to figure out ways of managing. And through the work of Damir Sudar and Michelle Netterloff is summarized in the next few slides. And I just want to highlight that because it's been a big effort to try to manage those. And we principally used the Omero database to allow us to store both the data as well as the metadata. That's all in-house. Damir has also been working, and Michelle have been working quite a bit to try to generate publicly available interfaces that allow us to actually sift through these data. At this point, we're up to about 10 terabytes of data and over 500,000 images that we've generated. As of, this is actually a few months back at this point, where our numbers are creeping up every month. But trying to figure out how to understand these data, it turns out it's great to be able to quantify them and extract image features. It's also incredibly useful to actually just take a look at them to interpret what's going on. And so this Links Clarion website gives a few different ways of doing that. This you actually could go into right now and surf around a little bit to see some of the data in there. But within here, what we've done is to create, let's see if I can get this here we go. Using Omero and Zegami, it's possible to actually sift through these data, looking at both the images as well as the image feature data, so that you can sort down to look for particular conditions of interest based on what the treatments were, or based on what the image features themselves were. This here is Omero, where it's possible to actually pull up individual images that are high resolution images. You can render them in different ways, split out the channels, and so on. Additionally, there's a bit of a simpler interface that allows you to choose which particular ECM or cell line or ligand combination you would like to see, and then pulls that up for you and does relatively simple displays of the data. But this as well is quite useful for just doing data mining. And then finally, this last one, the MEMA board, was done in collaboration with students from Avi's group. This one actually uses one of the tensor algorithms and actually pulls in all of the image feature data, and again allows data mining. So what I'd like to do now is step into a bit of the biology that we have really learned from using this sort of platform. And the first use case I will tell you about is a little bit about is the observation that the signals from the microenvironment strongly influence response to therapy. And a particular example that I'll give you is from HER2 breast cancer. We know that HER2 positive breast cancers are exquisitely sensitive to the HER2 inhibitor lapatinib. This we see both in the lab as well as in the clinic. What we have found is that response to HER2 is very much modulated by the environment in which cells are growing. So shown here in these images is the HER2 positive breast cancer cell line A565. We see within one of these images, each blue dot is a DAPI stained nucleus. On the right-hand side, the red dots are EDU nuclei with EDU incorporated. That EDU incorporation indicates that the cells are actively proliferating. These are cells in two different environments, shown in the white, PDGFAB or else neuregulin 1. In both cases, these cells have been exposed to lapatinib. So we see on the left-hand side, they are responding. There's nothing pink. They are fully responding without proliferating. In the context of neuregulin, these cells are actually still proliferating, ignoring the fact that we have dumped lapatinib inhibitor upon them. And if we quantify this after looking at 56 ligands, we see for most ligands, what you're looking at here, 56 ligands across the X, proliferation rate across the Y-axis. Most of those bars are very, very low, right, indicating that these cells are responding well to the drug. There's a few cases where the bars are actually very tall. Neuregulin is a huge outlier where there's just this incredible proliferation despite the fact that we have drug on board. This to us is incredibly important to think about in terms of, again, what is the environment in which a cell in the patient's body is living in, and how might we think about using that information to better design effective therapies. If we look at other cell types, in this case, HCC 1954, we find other environments that are permissive in the presence of lapatinib. These two particular cell types have different transcriptional backgrounds. We believe this is the reason for the difference in the microenvironmental response. And using I won't go into it now because the paper is about to come out, but using a lot of different integrative cell biology approaches, what we've done is to identify these particular environments that mediate resistance to HER2 inhibitors. And that suggests alternate combination therapies that may be most effective for patients with these two different types of diseases. HER2 positive basal-like disease versus HER2 positive luminal-like disease. More recently, what we've done is to really think about how microenvironmental signals may modulate the ability of cells to self-organize. What I'm showing you here are example images from two different primary mammary cells. 240L, which is cells derived from a young 19-year-old patient, and 122L, which are cells derived from a 66-year-old patient. In both cases, these are normal, healthy cells. And what we see in the left-hand side here is that these cells actually self-organize into this bullseye design. And this indicates an organization of epithelial and myoepithelial cell types. We see this phenotype only in this young sample and not in the older sample, indicating that there may be an ability, the ability to self-organize actually diminishes with age. It turns out this ability to self-organize is also associated with the environment in which a cell is grown. So what I'm showing you here, again, are from that same cell line, 240L, where we do see many cases shown on the far right-hand side there, where these cells are incredibly organized with this epithelial, myoepithelial organization. And then other conditions where cells are far less organized, shown on the left-hand side, live 1, where we see actually both the morphologies as well as the expression of the markers is quite different. We can actually quantify these sort of state and see that across the different environments, in this case, stratifying across the ligands, we see a very striking variation in the ability of cells to organize, indicating that this high-level phenotype is very much controlled by the environment in which cells are grown. We're exploring this further, starting to use the sort of deep learning approaches that Anne mentioned yesterday in her talk. If we're able to classify these images into organized versus not organized cells, apply convolutional neural networks, and in order to classify the images and try to better understand what are the features that are associated with this sort of organization, with the idea that that may help us ultimately to understand what is the relationship between some of these different environmental conditions and the phenotypes that they induce. What I'd like to spend the rest of the time upon is a project that we are embarking on. This is a links-wide collaborative project, actually, at this point, one that is really focused on trying to understand the influence on the microenvironment when thinking about diverse cell types, or diverse phenotypic endpoints, rather. What I'm showing you here are data from 2 different MEMA screens on the MCF10A normal-like cell line. And what you're seeing are, across the x-axis, data from 56 different ligands. Across the y-axis is cell count. If we focus just on the top set of box plots, what you can see is that the number of cells, or the growth rate in of cells, varies as a function of what the environment is and what ligands those cells are being exposed to. And in the top case, these are cells growing without EGF in the media. We find there are certain ligands that strongly induce proliferation. So on the far left-hand side, alpha-regulin, you see EGF epigen coming up as top hits. In the lower box plot, we see that there are ligands that actually reduce cell proliferation. We've taken this a bit further, and also in the image data extracted out the differentiation markers and found that across the MEMA screen, the environment in which cells are grown also strongly modulates the expression of differentiation markers. So on the left-hand side, we have examples from EGF. On the right-hand side, an example of the ligand oncostatin N. In both cases, we see cells are grown in the same, on the same substrate. The only variation in this case is the addition of the ligand, and we see a very striking difference in the expression of a keratin marker, keratin 5 basal-like marker. This tells us that the differentiation state is strongly modulated by the environment as well as cell morphology. And here's a set of images from the top 6 ligands that we now have started studying systematically in this particular project. In the top left-hand side is the PBS control. The other 5 images are from ligands identified as those that strongly influence various phenotypes. So from cell count, proliferation, differentiation state, and so on. It's the incredible thing about these sorts of images is that it was possible in the lab when running these experiments to simply look in the well and actually know what the treatment was, because these phenotypes are so very different in terms of how the cells grow. You see in oncostatin M, there's this very clumpy phenotype, whereas in TGF beta 1 in the middle there's this much more diffuse distribution of cells. When the only thing we're changing here is the ligand addition. What we've also found is that all of the data that I've shown you so far are single time point movies, or single time point data sets. What we have started doing as well, this is work led by Sean Gross in my lab, has really pushed me into doing some live cell imaging. And here are example images from 4 of these conditions, again with the PBS control on the upper left-hand side. And what we can see is that over the course of 72 hours, these cells actually are incredibly dynamic in their behavior and in their responses. And in fact, the phenotypic response varies quite a bit in terms of the migratory potential of these cells. So for instance, I want to draw your attention to the oncostatin M condition in the lower right-hand corner there, where what we see is that these cells end up with this migratory phenotype that is quite striking in terms of them creating these large colonies that move all around the bottom of the cell culture dish, and they go seeking out neighboring colonies. This sort of behavior we think is incredibly important in the context of metastatic cancer. We know that in order for a tumor to become metastatic, it must actually migrate off of the primary tumor and in order to get out to a distant site. And so this sort of condition is one we're really thinking about the biology underlying this particular ligand treatment. So in summary, what we found is that across this set of ligands that we've identified as being top hit in terms of their ability to modulate different phenotypes, we see a striking variation in how, in the number of phenotypes that these modulate, as well as the effects that these different ligands induce. And really what we see is that across these different treatment types, there's not a single ligand to phenotype association. Instead, we see a much more complex relationship between the different perturbations as well as the phenotypes that they induce. And so this sort of preliminary data is something that we are now working hard to try to understand the molecular basis of these sorts of observations. And the sorts of questions that we're poised to go after now are to ask, what are the regulatory, transcriptional, and proteomic networks that control these phenotypes? How are the different cellular endpoints related? And what are the rules that govern the integration of these diverse signals from the microenvironment to modulate these different phenotypes? In order to go after these questions, what we've done is to embark on what we're calling the NCF10A Dense Matrix project. This project is led by our center, but involves collaborations with all of the other linked centers as well. And what we've done is really try to leverage the power of the LINCS consortium in order to use all of the assays available in LINCS to try to understand all of these different molecular levels that are associated with the phenotypes that I just described to you on the previous set of slides. So going from the cell behavior, so this sort of live cell imaging assay, looking at protein expression with cyclic IF and immunofluorescence, as well as the RPPA, which is a reverse phase proteomic array assay. And then looking at transcriptional and epigenomic changes down at the bottom there. And the idea is that an integrative approach will really allow us to best understand how these different phenotypes are manifested. Additionally, what we will be doing, really this was very much encouraged by the dynamic responses that we saw in the live cell imaging, is to also be, when we run these assays, to look at several different time points out to 48 hours so that we can get a sense of how the molecular responses are changing over time. And all of those data are going to be available for all of the analytics that you heard about yesterday from Avi and Mario, so really using a lot of what's already available in LINCS. But additionally, I have a collaboration going with Mark Firstwhistle, who is part of the Detox Link Center, who's developed a lot of mechanistic modeling approaches for these sorts of dynamic data that we'll be generating. And so we're really hoping to leverage that approach to try to understand the cell signaling in the data set that I described previously. So in summary, what I've told you about a little bit about our center is that we've generated and released several large-scale imaging data sets to the public cloud, that the MEMA platform that we've developed enables discoveries about microenvironmental biology, and we found that the microenvironmental-mediated drug resistance, that the microenvironment modulates the ability to self-organize, and we also see striking effects on growth, differentiation, and motility. And finally, this NCF10A project will explore the molecular basis of complex phenotypes. And with that, I'd just like to thank those at our MEP Link Center listed here, and in particular, Sean Gross, who I've highlighted, who really has been spearheading a lot of the NCF10A effort. And with that, I'm happy to take questions.
Thank you, Laura. Yeah, we can take 2 or 3 questions.
How stable are your microenvironmental arrays? Can you store them? Can you transfer them between labs? And then a little bit related question: can you spot them down onto matrix sequence?
So they are quite stable. We can they get desiccated and stored for several months. We have sent them off to collaborators where then they can culture their own cells and image them. So that's quite straightforward. We are also thinking about printing onto other substrate thicknesses. This is something Mark Labarge, who is one of our collaborators in MEP Links, has a strong interest in substrate stiffness and how that modulates cell phenotype. We know this is a very important factor both in normal tissue as well as in context of tumors, right? A tumor feels like a lump because it is incredibly stiff in terms of the extracellular matrix proteins. So I think it is possible to do that. It's not something that we at our center have done, but Mark certainly is doing this in collaboration with us.
So thank you again so
Thank
much,
you.
Laura.